Hi, I'm Beth Pitel. I'm a senior developer at the Mayo Clinic in the Clinical Genomics Laboratory, and I'm excited to share with you an overview of some resources that I find particularly helpful in facilitating cancer variant interpretation. I have nothing to disclose. The objectives of this talk are to help you understand the types of resources that are available for variant interpretation, to review some of the more prominent cancer genome interpretation resources, and to understand the landscape of interconnectivity between these resources and what that means for our field. So when you're looking at large assays with high throughput, there are automatically processed portions of these assays that lead to this bottleneck of variant interpretation. So right now we're able to automatically process, annotate, and filter variants using bioinformatic pipelines and algorithms, but ultimately this bottleneck occurs when the human element needs to be integrated into this workflow. And so that person needs to actually interpret all of that data that comes down to this bottleneck in the interpretation pipeline. And that person is responsible for putting together a report for the clinicians. So I find it particularly helpful to understand the difference between data and knowledge, or information that you would find in a database or data portal versus information you would find in a knowledge base. So I like to think of data as what you would see in a results section of a manuscript. And this might include statistics that are derived algorithmically from large data sets, for instance, the top 20 mutations in a specific cancer type. Knowledge, on the other hand, I like to think of as more of the discussion section of the manuscript, where we're critically processing the data for clinical relevance. Often knowledge bases contain information that's populated by dedicated curation staff or expert crowdsourcing. And these resources are often driven primarily by scientific literature. And then there's this area, the gray area in the middle of the screen, that is bridging the gap. So you can use data to automatically make some inferences about what that might mean in a clinical setting. And so databases often have really great visualizations, and that can help you understand what that data means. So these might include a 3D structure of the proteins that you can use to understand where your variant lies in the protein structure relative to maybe a drug target. You can create survival plots with some resources, or mutation grids where you can look at co-mutations. Some databases incorporate pathogenicity scores and algorithms, and some have available informatics workflows that you can use to help classify variants. So this slide is meant to just summarize the different kinds of resources that are available and the different kinds of data that are available. So we have literature search engines, like for instance, Genomics Mastermind or PubMed, Google Scholar. We have clinical experience databases like ClinVar, cancer variant databases like Cosmic or CBioPortal, in silico predictors like Civic or Polysen, cancer variant knowledge bases like Civic or Oncology, clinical trials resources, population databases like Nomad, or 1,000 genomes, or cytogenetic knowledge bases like the Middleman database or Atlas. And what's happening in our field right now is that we have a lot of this aggregation activity where a lot of different data types are now being kind of packaged together or being connected with each other through API or citing each other. in the resources. And so this happens in a couple different forms. It happens in the form of publicly available or commercial products that are now integrating these different types of data into a single piece of software. This happens by harmonizing knowledge. So I'll talk in a little bit about a group that is taking information from several different resources and then standardizing the annotation so it's more readable. And you can search that information with a single query. And then also you have molecular tumor boards, which different people can use different resources and bring that knowledge back into the tumor board, either in person or virtually, or however you do tumor boards these days. And so in the next several slides, we'll be reviewing a number of different resources, and I've coded them with these icons for ease of recognition. So some of these resources have information at the gene or variant level. Some have information at the cytogenetics level. Some contain evidence for clinical relevance of each variant. Some contain exemplary visualization tools. Some are manually curated by experts in our field. We've talked a little bit about a knowledge base versus a data portal or database, and I'll make those distinctions on these slides as well. Some resources have data available for download, and some resources have API available, which helps you connect these resources in an automated way to other applications. So COSMIC, or the Catalog of Somatic Mutations in Cancer, is the first resource that we'll go over. COSMIC has so much functionality, it's really hard to summarize this in just a couple slides. So it's really fun to go and play around in COSMIC because they're always coming up with something new, and they're really a leader in thematic data. So this is developed and curated at the Wellcome Sanger Institute in Cambridgeshire, UK. There are currently over 10 million mutations curated on this database, and they're currently on version 93 of the database. You can just imagine the amount of effort that has gone into curating and building this repository. They have a very famous list called the Cancer Gene Census. You've probably heard of it or heard of people using it in different ways. They also have a cancer mutation census, which I'll go into in the next slide. COSMIC 3D is a great tool, and this is where you can see a 3D model of the protein. Also, there are mutational signatures available on COSMIC, annotations across genome builds. So if you have data from HG19 and 38, you are able to see both of those annotations. There are downloads for licensed users, and brand new at COSMIC are actionability data that you can download with a license as well. In this panel, I wanted to show the utility of the cancer browser, and in this case, I've filtered by tissue, subtissue, and histology. So we filtered by the tissue being central nervous system, brain tissue, and this is a atypical teratoid rhabdoid tumor. And we see our top 20 genes that are mutated in this cancer type. And generating a list like this can be very useful when you're running quality control on a panel or a gene list that may be created in-house, and you wanted to make sure that you capture a lot of the genes that are clinically relevant or often mutated in that tumor type. And so this might prompt additional literature searches or investigation into genes that maybe you didn't consider initially. And I really wanted to show kind of on a gene level what COSMIC can do. So I have here the ABL1 gene in the COSMIC 3D model of the protein. Also, there are several genes that are annotated for hallmarks of cancer. And the hallmarks of cancer are really kind of biological functions of cancer. And the genes that are annotated for the hallmarks of cancer are marked as either promoting or suppressing these biological activities. And then I also wanted to highlight the mutation census. So the mutation census actually catalogs all of the mutations that are in COSMIC database in a kind of special way here. So the mutation significance, which I've shown kind of on the bottom of the screen here, classifies variants into different tiers or significance levels for those mutations. Also in the mutation census are ClinVar entries, nomad frequencies, evolution probability, frequency of the occurrence of the mutation in COSMIC, and conserved sequences. The next database I'm going to go over is CBioPortal, and CBioPortal is developed at Memorial Sloan Kettering and hosted by the Center for Molecular Oncology. CBioPortal houses data from lots of large studies, including TCGA and many others. The information in CBioPortal is easy to view and easy to stratify. So I really encourage people to play around in CBioPortal, especially if you have like a disease interest, and work on manipulating that data within this interface, because it's really pretty cool. Also, CBioPortal in the last year has posted several tutorials on YouTube, so you can go check those out. And CBioPortal for years has allowed us to visualize our own data as well, which I'll show on the next slide. So on the bottom panel here, I have looked into a specific gene, and when you search for a gene in their quick search function, you can see the alteration frequency and also the alteration type present in several of these large studies. So it really gives you a sense of the importance of that gene and in what cancer types it is altered. So I did mention that you can visualize your data in CBioPortal, and one of the ways to do that is by using the Mutation Mapper. And what the Mutation Mapper does is allows you to visualize your data in a lollipop plot like this one. And then the other way to visualize your data in CBioPortal is by using the OncoGrid. And the OncoGrid is really nice if you want to stratify your data based on specific molecular profiles, or if you want to view co-mutations. This is a really helpful way to do that. OncoKB is the knowledge base partner to CBioPortal, and they're both hosted out of Memorial Sloan Kettering. OncoKB is probably most well known for its annotation using evidence levels for therapeutic significance. But recently, they have included evidence levels for diagnostic and prognostic evidence as well. The prognostic evidence is currently available for hematologic malignancies only, but solid tumors will be coming, I'm sure, fairly soon. Also, if you would want to annotate your own lab data using OncoKB annotations, there is a way to do that as well. And so at the bottom of the screen, I have shown the homepage for OncoKB, and it's really pretty slick. You can toggle between the therapeutic evidence levels, diagnostic levels, and prognostic levels to see the representation of these data types in the knowledge base. And you can also do a quick search for a gene name or disease type right in that search bar. If you search for ATM, you'll get the bar graph that's in the top right of the screen, and you can see the percent of samples with alterations in ATM across tumor types, which is also very helpful. And I really wanted to include this slide as well. This is available on OncoKB website as well. But I think it's really helpful for those of us who work in a clinical setting to be able to align these evidence levels with the AMP-ASCO-CAP guidelines. So this is really handy that they've kind of made those connections for us. So the next resource I'm going to go over is Civic, or the Clinical Interpretation of Variants in Cancer. It's hosted out of the McDonald Genome Institute at WashU in St. Louis. They have a really great curation team at Civic, but they also include crowdsourcing from experts in our field. They have several YouTube tutorials that not only teach you to use Civic, but also teach you how to contribute if that's something you're interested in. They've recently had a key collaboration established with the ClinGen Somatic Cancer Domain Working Group. And what this means is that this working group from ClinGen will actually populate the Civic database with their findings. And so I think that this is a really cool collaboration that will make sure that a lot of the prominent somatic variants are in Civic. Also, they've had a recent integration with Protein Paint, which is part of the St. Jude's Pecan data portal that I'll talk about on the next slide. So I wanted to show a couple of screenshots from Civic. I find the browse function to be really helpful in Civic. And so when you go into the browse function, you can type into any of the search bars towards the top of the table. Here I've typed in cholangiocarcinoma, and what populates below are all of the variants that are associated with cholangiocarcinoma in Civic. So this is a really helpful way to, again, kind of do that cross-checking QC review of variants in a specific cancer type. And there are several other ways to use this as well. And also in Civic, one of the really cool things about Civic is how they display evidence. And so this is one of the evidence tables displayed for an EGFR amplification. And so in the table, you'll see 4 rows, and these represent 4 different pieces of primary literature. And the icons in the table are coded for recognition. So you can see this first line actually means that it supports a poor prognosis. And then the next 3 lines, you'll see drugs listed in the drug column in the table, and these are related to more predictive or therapeutic implications. But when you actually click on one of the rows or one of the pieces of evidence, the panel below this populates where you can see a 1 to 2 sentence summary of this piece of evidence and link out to PubMed to make your own assertions, which I always recommend. The Pediatric Cancer Data Portal is developed from St. Jude's Children's Research Hospital. And I think that they're a real leader in visualizations and just visualizing large sets of data in general. In addition to the visuals we'll go over today, I wanted to make aware that this group also has some publicly available bioinformatics workflows, including the Pecan Pi, or the pathogenicity information exchange, which will classify variants into different metal levels. So on the right side of this slide at the top is a representation of all of the cases that are cataloged in Pecan data portal, and they're stratified by disease type. So the yellow is solid tumor, the blue is brain tumor, and the red is hematologic malignancies, and then different subtypes within those categories. Also on the home page, you'll see a word cloud with different gene names, and you'll see how frequently these genes are altered within those disease types, kind of color-coded the same as the pie chart above. Another really powerful visualization tool in Pecan Data Portal is Protein Paint, and that's shown below. The only difference between Protein Paint and Genome Paint is within Protein Paint, you'll only see the mutations that fall within exons or coding regions for fusions. With Genome Paint, you start to see some of those more intron-related mutations, and you'll actually look at a larger transcript of the gene itself. So below is the APOL1 gene by Protein Paint. So you can see the lollipop plot there. You'll also see buttons that connect not just the data from Pecan Data Portal, but also Cosmic, ClinVar, and Civic, which I referred to earlier. So probably some of my favorite things about this graph are that you can see the domains that are color-coded on the protein itself and on the bottom rectangle of the protein. And then also, if you click on a fusion, you'll actually see a representation of the pertinent fusion and understand which domains from both fusion partners are in the fusion transcript, in the actual fusion that was observed. So the Jackson Clinical Knowledge Base, and this one, actually the other animation went a little too quick, but we can talk through it. This is based out of the Jackson Laboratory, and there are several different there are a couple different licensing tiers I wanted to mention here. There's the CKB Core, which is the free license to JAK CKB, and this includes kind of the basic, primary driver genes for cancer and a lot of the therapeutic information that is in JAK CKB. The Boost really ups the functionality. It has a larger gene list that the knowledge base covers, and then also adds some of these more interactive, visual, kind of stunning pieces within JAK CKB. And then the third license that we can't see right now is CKB Flex. And the CKB Flex allows for data downloads and kind of more interactivity between the CKB and your own lab applications. And then in addition to a lot of more therapeutic, detailed information that you find in JAK CKB, they have this concept of a molecular profile that is unique to JAK CKB. And what this means is that you can actually look at information regarding not just one variant, but a combination of variants. And so in one of the panels on this slide, you'll see that we have searched for an ALK rearrangement and a MET amplification. And we were able to find information on this molecular profile and linking to two pieces of primary literature. And then in the front of the slide, it's a sunburst plot that's available through CKB Boost. And this is annotating or representation of all the annotations in JAK CKB in relation to the ALK genes. So you can further filter your search through an interactive sunburst plot, which is exciting. The next resource I'll go over is the Molecular Oncology Almanac, and this is from the Broad and Dana-Farber, and you can search by biomarker, cancer type, level of predictive implication, or therapy. And the level of predictive implication might be my favorite thing about this because you're so easily able to identify the pieces of evidence that make different alterations a tier 1 event. So you can find your pieces of evidence that that make a gene and therapy combination FDA approved. Like you see on this bottom panel, the top line, you'll see that TSC1 oncogenic mutations with everolimus. There's a link to the evidence saying that that is an FDA-approved therapy for TSC1 oncogenic mutations, which is really, really helpful. So the Middleman database of chromosome aberrations and gene fusions in cancer is funded by the National Cancer Institute and a couple of Swedish cancer societies. It's supported by the Institute for Systems Biology Cancer Genomics Cloud, and it's one of the few cytogenetics related databases, knowledge bases out there. And so on this database, you can search over 70,000 cases that are curated from the primary literature. And you can download the entire database, or you can download results from a search. Also, I've heard that there's some visualization coming soon, so stay tuned. Probably my favorite search function within Middleman is the recurrent chromosome aberration search, where you can search for a chromosome band or a particular imbalance, and it will search the literature and it'll find cases with that same alteration. And you can link to the primary literature. You can see the karyotype for those cases along with the diagnosis or morphology. So it's a pretty cool resource. Genomics Mastermind is a literature search tool that uses natural language processing to mine and prioritize over 30 million articles and over 2 million supplemental data sets. There are 2 licenses with this product as well. There's a basic license, which allows you to run a simple query, or a professional license, which adds a lot of functionality and a lot of kind of more in-depth searches. And so you can explore associations, which I'll go into in the next slide, or focus view in the pro version. So below, in the panel below, you'll see a graph with a bunch of circles in it. And each circle represents a publication. And on the y-axis is the citations per journal, and on the x-axis is the year. And the size of the circle corresponds to the impact factor of the journal. So this is for the axon 2 gene. This next graph is also on their user interface, and it allows you to refine your search to a particular gene domain or a particular residue within that gene, which is pretty helpful. And I'd mentioned that there's a new association function in Genomics Mastermind for the pro version of Mastermind. And what you can do here is really adjust your search criteria to include so many things, including CNVs, therapies, diseases, variants, and then you can look at all of your evidence once you've kind of taken all the pieces that you want to refine in your search. And I really wanted to touch on a little bit of data harmonization by talking about the Variant Interpretation for Cancer Consortium. This group is a driver project and part of the Global Alliance for Genomics and Health. And what this group did was they took several different prominent knowledge bases and took the elements from these knowledge bases and annotated them similarly so you could query multiple knowledge bases with one search. And this is really important. The need for this arose from interrogation of all the variants within these knowledge bases, and we found that most variants were only annotated in either 1 or 2 knowledge bases, and only 8 variants were annotated across all of these knowledge bases. So that was really surprising. So when I first gave an iteration of this talk back in 2017. Different knowledge bases and databases were starting to make connections with each other through API or through data sharing or by referencing each other in the knowledge bases themselves. And over the last couple of years, we've seen these connections become more intricate and become more involved with each other. So we've seen more integration between different databases and knowledge bases. And aggregation activity just furthers this integration. So we're not there yet, but what we're hoping for is this ideal state where a user will be able to define what they want in a comprehensive resource and be able to tap into everything with one interface instead of having to go into 10 or 12 different resources to work up a variant. So in summary, cancer genome interpretation and research require the right resources, and the landscape of genomic resources is constantly evolving and connecting with each other. It's a really exciting time for genomic knowledge bases and databases, and we can all benefit from the collaborations that are being made. And I highly recommend, if you have the bandwidth or if you have the interest, to connect with groups like Civic or ClinGen or any of the knowledge-based groups to help further their efforts, because it's really a neat field to be in. And then looking into the future, we can expect to see a more connected landscape of resources, aggregation activity, and just overall comprehensiveness of these resources as they become more connected. And then we'll also look forward to seeing further development and incorporation of things like natural language processing, machine learning, artificial intelligence into these resources as well. So I have a lot of people to acknowledge. Thank you to the Lab Roots Committee for inviting me to come talk today. Thanks to the Genomics of Oncology Annotation team at Mayo Clinic for supporting this work and really driving these efforts. Also thanks to the Cancer Genomics Consortium for their ongoing support and collaboration as well, and all of the knowledge-based experts that have contributed to my understanding and my really nice experience with all of these resources. So thanks so much for listening to me today. Feel free to email me with any questions, find me on Twitter or LinkedIn as well, and have a great time during this virtual event. Thanks so much.